Dear viewers, greetings. In this video, we are going to see about the spirochete Treponema pallidum. First, general characteristics of Treponema pallidum. Treponema pallidum was formerly known as Spirocheta pallida, and it is a gram-negative, thin, spirally coiled spirochetes, and it is motile in nature and shows corkscrew motility. The size of Treponema pallidum is ranging from 6 to 15 micrometer in length and 0.2 micrometer in diameter. It belongs to the family Treponema tarsinae, and capsules absent, endospores also absent. Regarding mode of respiration, Treponema pallidum is microaerophilic in nature and it requires very small amount of oxygen for its growth. It cannot grow on standard culture medium and its optimum temperature is 30 to 37 degrees Celsius and optimum pH is 7.2 to 7.4. Regarding the habitat, it is an obligate parasite, the meaning that it cannot exist outside the host because it lacks the ability to synthesize essential nutrients and consequently needs to obtain them from the host. Regarding the discovery, Treponema pallidum was discovered by Fritz Schroding, who was the German zoologist, and the German dermatologist Erich Hoffmann in the year 1905. Disease transmission of Treponema pallidum. Treponema pallidum infection are mainly transmitted by direct contact with skin lesions, mucous membranes, body fluids and secretions like semen, vaginal discharge, saliva and blood of infected person during the sexual contact. The second mode of infection is rarely by kissing and blood transmission, and finally transmission by congenital transmission, that is the vertical transmission from mother to child. Virulence factors of Treponema pallidum. The ability of the microorganisms to cause an infection is called as virulence, and the term virulence was first coined by the scientist Louis Pasteur. Virulence factors are the factors which are responsible for causing infection in human beings. Here, the Treponema pallidum have three virulence factors. The first virulence factor is outer membrane protein. The second virulence factor is the enzyme hyaluronidase, and the third virulence factor is coating of fibronectin. The first outer membrane proteins helps in the adherence to the host cells, and the enzyme hyaluronidase helps in perivascular infiltration, that is the macrophages and mast cell infiltrate around the blood vessels, and finally the coating of fibronectin protects the Treponema pallidum against the phagocytosis or cell eating process. An incubation period of Treponema pallidum is ranging from 10 days to 3 months, and the infective dose required to cause an infection by the Treponema pallidum is variable. Pathogenesis of Treponema pallidum. Treponema pallidum is the causative agent of syphilis, a chronic infectious human disease transmitted between the individuals via sexual intercourse or from mother to child. Untreated syphilis progresses in a series of distinct stages like primary stage, secondary stage, latent stage and tertiary stage. Infection is initiated when Treponema pallidum penetrate dermal microabrasions or intact mucous membrane, resulting in the primary syphilis. Primary syphilis usually presents itself as a single painless ulcer at the site of infection, and the secondary syphilis occurs approximately 3 months after infection and presents itself with a variety of symptoms, most notably lesions of the skin and mucous membrane. After several months, secondary symptoms will disappear. This is called the latent phase. If untreated, latent phase may progress to tertiary phase. Tertiary syphilis does not manifest until years after initial infection and can affect many different areas of the body. Tertiary syphilis can cause destructive lesions on the skins and bones, which are usually benign, and the more deadly manifestation of late syphilis affects the cardiovascular system and the central nervous system, causing infected individuals to experience insomnia or difficulty in sleeping and changes in the personality. Clinical diseases caused by the Treponema pallidum are primary syphilis, secondary syphilis, latent syphilis, tertiary syphilis and congenital syphilis. The first clinical disease caused by the Treponema pallidum is primary syphilis. Primary syphilis is the stage of infection which occurs 3 to 90 days after infection. A painless source appears at the site of infection like mouth, anus, rectum, vagina or penis. These are called as cancres. The sores heal on their own after 3 to 6 weeks but can still spread syphilis. It is easily treated and cured with medicine. The fact that this ulcer heals spontaneously within 2 months gives the patient a false sense of relief. Secondary syphilis. The clinical evidence of disseminated disease marks the second stage of syphilis. In this stage, patient typically experience flu-like symptoms with sore throat, headache, fever, myalgias or muscle aches, anorexia, lymphadenopathy or swollen lymph node, and a generalized mucocutaneous rash. The flu-like symptoms and lymphadenopathy generally appears first and then are followed a few days later by the disseminated skin rash. The rash can be variable like macular, papular or pustular, covers the entire skin surface including the palms and soles, and may resolve slowly over a period of weeks to months. Raised lesions called condylomata lata may occur in moist skin folds and erosions may develop in the mouth and on the mucosal surfaces. The rash and symptoms resolve spontaneously within a few weeks and the patient enters the latent or clinically inactive stage of the disease. Latent syphilis. During this latent syphilis phase, the syphilis bacteria are still alive in the body but asymptomatic. Affected patients are contagious during this stage, but syphilis may still affect the patient's heart, brain, nerves, bones and other parts of the body. This phase can last for years. Not everyone who has syphilis will enter this phase of infection, as some people will go into the tertiary phase or tertiary stage directly. Tertiary syphilis. Tertiary syphilis begins when symptoms from the secondary stage of syphilis disappears. Syphilis infection has started to affect all the organs. This can lead to death. There are three symptoms of tertiary syphilis. The first one is devastating destruction of virtually any organs or tissues. Example, arthritis, dementia and blindness. The second symptom is granulomatous lesion may found in bone, skin and other tissues. The third symptom of tertiary syphilis includes increased incidence of neurosyphilis. Finally, congenital syphilis. Congenital syphilis infection can lead to serious fatal disease resulting in latent infections or asymptomatic infection, multi-organ malformations or death of the fetus. Most infected infants are born without clinical evidence of the disease, but rhinitis then develops and is followed by a widespread discriminating maculopapular rash. Teeth and bone malformation, blindness, deafness and cardiovascular syphilis are common in untreated infants who survive the initial phase of the disease. Laboratory diagnosis of Treponema pallidum. The first diagnostic method is microscopic examination. Direct fluorescent antibody microscopy is very useful if mucosal ulcers are observed in primary or secondary stage of syphilis. In motility test, motile spirochetes which shows corkscrew motility are observed under the dark field microscope. This image shows the morphology of Treponema pallidum under the direct fluorescent antibody microscopy. Next, serology or serodiagnosis. Serology is very sensitive in secondary and latent stage of syphilis. Some of the non-treponemal tests used for the identification of Treponema pallidum infection or syphilis are venereal disease research laboratory test or VDRL test, rapid plasma reagent test or RPR test, unheated serum reagent test or USR test, toluidine rate unheated serum test or TRUST. And some of the treponemal tests used for the diagnosis of syphilis are Treponema pallidum heme agglutination test or TPHA test, fluorescent treponemal antibody absorption test or FTA-ABS, and finally enzyme immunoassay or EIA. Finally, molecular diagnosis of syphilis. 16S rRNA sequencing and polymerase chain reaction are used for the molecular diagnosis of syphilis. Finally, treatment and prevention. The first effective treatment, that is the salvacin drug, was developed in 1910 by Sahachiro Hata, the Japanese bacteriologist, in the laboratory of Paul Ehrlich. It was followed by the introduction of penicillin in 1943. Penicillin is the drug of choice. Deoxycycline is administered if the patient is allergic to penicillin. Safe sex practices should be emphasized and sexual partners of the infected patient should be treated. No vaccine is available for the syphilis infection. Dear viewers, that's all about the spirochete Treponema pallidum. Thank you for your support. Thank you.